Good afternoon, everybody. Thank you so much for joining us today for the New England Wetland webinar series hosted by NUEPIC. My name is Audra Martin, and I will be your moderator today. I'm so happy to welcome our speaker, Sandy Crystall of the New Hampshire DES Wetlands Bureau. She's going to discuss New Hampshire's progress over the past several years to develop wetland-specific water quality standards. She is a training and research specialist in the New Hampshire Department of Environmental Services Wetlands Bureau. Sandy has spent more than 8 years working on wetland condition assessment and the development of wetland-specific water quality standards. She was the New Hampshire team botanist and regional trainer for the vegetation protocols used in EPA's National Wetland Condition Assessments back in 2011 and 2016. And in her prior 12 years in the Wetlands Bureau, she conducted outreach and was a permitting instructor. She has also held positions with FEMA, EPA, and in consulting. She's trained in biology and environmental planning and is a professional wetland scientist. In her spare time, she likes to botanize and do genealogical research. So we're so excited to have her here today, and thank you all again for joining us.
Oh, good afternoon. Thank you all for your patience. Thank you for the opportunity to share New Hampshire's experience and progress toward the development of biocriteria to assess wetland condition. I'm going to discuss how we started, describe this project and its results and considerations for next steps in the development of biocriteria. So since the mid-2000s, New Hampshire has taken various steps toward the goal of assessing wetland condition. DES conducted a level 1 analysis of its wetlands. The New Hampshire Natural Heritage Bureau started to use the Ecological Integrity Assessment, and in 2012, DES with the Natural Heritage Bureau applied several wetland assessment methods to more than 30 wetlands. DES has drafted a wetland monitoring strategy and a plan to develop wetland-specific water quality standards. What emerged from the development of the plan for wetland-specific water quality standards were discussions with Maine DEP biomonitoring staff, Jean DeFranco in particular, who had presented to the Wetland Water Quality Standards Subcommittee. So Jean had described how the biological condition gradient approach, the framework that describes incremental changes in aquatic systems, was reflected in Maine's tiered aquatic life use approach legislatively and, of course, in their biomonitoring. DEP was working on, actually had developed a linear discriminant model. At the time, it was based upon 12 macroinvertebrate variables shown here. The model requires a generic diversity of 15 and a total abundance of 50, and reports out by legislatively assigned water quality classes. Then Maine DEP had started to report on wetland condition in its 2010 305B report. So with support from Maine DEP, the EPA grant, DES decided to apply the Maine DEP macroinvertebrate-based protocols to New Hampshire's wetlands. So we'd apply the protocols of sampling macroinvertebrates, the rapid assessment, the water quality sampling. We provided our macroinvertebrate data to Maine DEP in the required format, and they would run it through their predictive model. Following the concept of using multiple biological assemblages to assess wetland condition, we also sample vegetation to enable application of floristic quality assessment metrics. We'd also apply the ecological integrity assessment as we had done before. So I want to take a moment to review the floristic quality assessment, which is a method based upon a coefficient of conservatism that is assigned to each plant species based upon the species tolerance for disturbance and fidelity to a particular plant community. A C value of 0 is assigned to non-native species, such as the common dandelion or Phragmites, a common reed. And a C value near the maximum of 10 is assigned to those species typically associated with a very specific habitat type, such as a bog or salt marsh. Species with intermediate tendencies are assigned C values in the middle range. Similar to the approach taken in Minnesota, where threshold C values for multiple wetland types in undisturbed and impacted conditions were developed, the New Hampshire Natural Heritage Bureau is determining thresholds, weighted mean C, one of the forest quality assessment indices, for our minimally and least impacted wetland system types. So the whisker and box plots shown here show the range and median of mean weighted C values for 14 of 25 New Hampshire wetland system types. The balance of those systems will be developed, will be completed later this year. I have labeled 3 wetland types and circled 2 of several wetland types that we sampled. You may notice that the drainage marsh shrub swamp at the left has a lower threshold, median 2.9, a C value of 2.9, than a medium level fen with its median C value of 5. It is likely that a bog in poor condition would have a higher weighted mean C value than an undisturbed drainage marsh shrub swamp, as Minnesota has shown. So supporting the concept that one value for all wetlands would not be appropriate to apply as a threshold. So we applied 2 rapid assessments, the wetland human disturbance assessment that Maine was using and our ecological integrity assessment. These include a desktop landscape analysis, level 1, along with the level 2 field-based observations, landscape features, hydrology, vegetation, and soils. Our target wetland type was open water wetlands, lacustrine, palustrine, and low gradient riverine fringing wetlands. These were identified with National Wetland Inventory map data, aerial imagery, and other sources. The assessment area included the open water and contiguous wetland types where no natural or artificial break was present. And this was following the ecological integrity assessment approach. The Ecological Integrity Assessment, or EIA, evaluates hydrology, vegetation, and landscape context and soil. The landscape context, which is the level 1 analysis, the GIS-based, includes a land use index, which is based upon a raster from land use from the National Land Cover data set, and on which the buffer zone of 100 meters, 250 meters, and 500 meters are overlaid. And then the distances are weighted, and the pixel values by land use type are weighted. The landscape context also includes a percent of perimeter having buffer, and that's a 10-meter line that's around the wetland. And we evaluate the intactness of that as well. And then an average buffer width is calculated where those 8 spokes that you see, kind of like a ladder there, we evaluate the disturbance along the 8 lines extending out 100 meters from the wetland. So the wetland human disturbance looks at watershed characteristics in terms of land use, as well as vegetative modifications, hydrologic modifications, and evidence to chemical pollutants in the wetland and adjacent upstream sources. The buffer that it uses is a 100-foot buffer around the wetland. So the macroinvertebrate sampling occurred, excuse me, where in these wetlands where water depth is 1 meter or less, samples. 3 replicates were taken in a wetland, so there were 3 ones replicate at each location. And a 1 meter measured sweep technique was used with a dip net. The macroinvertebrates were, sample was sieved and then preserved. Contract taxonomists processed, identified, and counted the specimens. And the taxonomic data was provided to Maine DEP for input to the model. So for water sampling, we used both field meters and took a grab sample. We took measurements with the field meters at the 3 macroinvertebrate sampling locations. Dissolved oxygen, pH, temperature, and conductivity, and specific conductance. This, in Maine, at least at the time that we started, they were taking these measurements at 1 location, and we felt that if we were taking the macroinvertebrates from 3 locations, we would do the meter sampling at the 3 locations. We also took 1 water grab sample at 1 of the 3 locations and analyzed it for the water parameters here: alkalinity, chloride, chlorophyll A, dissolved organic carbon, and various nutrients. We had, in the first 2 years of the project, under the first grant in 2014 and 2015, we had analyzed for dissolved orthophosphorus, but there were so few reportable levels that came out of that, and it was $40 a sample, that we thought that it wasn't worth the cost to get that limited data, and the lack of the data wasn't really telling us a lot. So the vegetation sampling, vegetation was sampled in 2 ways. For aquatic vegetation, we used a shrub rake at 3 macroinvertebrate sampling locations. We recorded vegetation in a 5 meter area around that location, which usually included shoreline vegetation, where it was more of an open pond setting. We also recorded additional species observed between sampling locations. We developed species lists, and so we could apply fluorescent quality assessment. Initially, we did more of a present absence. Later on, we developed a more detailed protocol at the same locations, but collecting additional information, but we didn't have it for the whole length of the project. So the predictive model results are shown on this location map. So we sampled 42 different wetlands, and the resulting attainment class is shown with the different color and shapes. For 10 wetlands, the indeterminate status was assigned because the model threshold for generic richness of 15 or total abundance of 50 was not met. The sum of the indeterminate results occurred, or I should say most, 8 out of the 10 wetlands with the indeterminate results were from the first 2 seasons of sampling, and were likely due to sampling issues, while the others, not all of the 8, but while the others were just the nature of the wetland. So what do these wetlands look like? So for predicted attainment class A, these tended to be less developed areas overall. The landscape and the buffers are pretty much undeveloped. The center wetland that shows the orange and red areas, the red areas are the land cover data set, the pixels for the developed roads land uses, and the orange are like hayfields and agricultural areas. The ecological integrity assessment actually has includes collapsing all the different national land cover data sets to 4 categories. So that's why you see 4 here. The bottom right buffer area, you can see there's a lighter green and a darker green. So those are 2 of the 4 categories in addition to the orange and the red ones. So for the predicted attainment class of B, we now see some wetlands that have more greater amounts of developed areas. At the bottom left is in Nashua, and that wetland is actually includes a wild rice marsh, which is quite amazing. The far right is Jaffrey, New Hampshire, where the developed area is actually at the downstream end of the wetland, which is to the north. So for predicted attainment class of C, these also have a range of some very undeveloped landscapes and some more developed landscapes. The same with the wetlands that were given an indeterminate status. 2 of those 3 wetlands that the left and the right are both in areas that are pretty much undeveloped and protected, and the middle one is in Salem, New Hampshire. So there's seems to be quite a diversity.
Want to interrupt? Sandy, we're seeing your Class B slide still.
Yeah.
So there may be a disconnect here.
I wonder whether it's just a real big delay.
Yeah, the slides are generally about 20 seconds behind. So you started talking about one new slide, and we're still looking at the older slide. So I've noticed a 20-second delay.
Yeah, Sandy, go ahead. Just keep that in mind.
Right. Well, I think for the B and C classes and the I indeterminate status, there's a real mix of some of the sites. Some of the sites have very undeveloped, undisturbed landscapes, and some of the wetlands have very developed landscapes, and they're in the same either attainment class or that indeterminate status. So it just, the A's seem to stand out as, okay, they're pretty much undeveloped, and that was perhaps reflected in the macroinvertebrates. It's unclear what's happening with the B and C, the wetlands given a B and C attainment status and the indeterminate status. Are you still on the B?
No, we're actually jumped to the indeterminate. So I think we're caught up.
Okay. All right.
Can I ask a quick question here?
Sure.
What is the role in determining the classification or the class status of non-native or invasive species? Are they a key factor in determining what status, or is it not really an issue in the samples that you have?
With the macroinvertebrates?
Mm-hmm. Oh, macroinvertebrates or the vegetation?
Okay, well, the status, the model status is based only on macroinvertebrates.
Oh, okay, okay.
We'll get to the vegetation part of things. Thank you, I didn't clarify that enough. But the attainment class is based only on the macroinvertebrates. Maine is also developing an algae model that they actually sample algae at the same time that they sample the macroinvertebrates. New Hampshire chose not to do the algae and then chose to do the vegetation. And in fact, we actually shared our vegetation protocol with Maine, and so they have actually the past few years been using our vegetation protocol, but their algae model was a higher priority for them to work on, so they've not looked at their vegetation data from using the same protocol, or similar protocol, I should say. Okay, so we'll get to more of the vegetation discussion in a minute. And I'll take it slow. Okay, so this next slide, which will show up eventually, is box plots of the data collected from the two rapid assessments, the ecological integrity assessment and the wetland human disturbance assessment. And the box plots provide some indication of the metric or the method by attainment class or indeterminate status. When we analyzed our data, almost all were non-parametric. And I'll get into some of the other things we did find. Is the plots visible to folks?
Yes.
Okay, good. All right. So for the rapid assessment, the EIA, the highest score, the higher the score, the better it is. For the wetland human disturbance assessment, the lower the score, the better it is. I didn't kind of normalize them. But the median EIA score for wetlands with a predicted attainment class of A was among those statistically significant results. So there was so between A and B, it was statistically significant for the EIA. So once again, the attainment class of A seems to stand out apart from the other classes. This next set of whisker and box plots is the floristic quality assessment data. So we analyzed the mean C by attainment class, but the range of values seemed to overlap between the attainment classes. There wasn't anything, the attainment class A did not stand out. We looked at some other floristic metrics. While not statistically significant, the median percentage with a C value of 0, so those were the non-native species, with a predicted attainment class of A is visually different when compared with the medians for other attainment classes or indeterminate status, but it wasn't considered statistically significant. In the third plot, the wetlands with an attainment class of A had a much higher percentage of the species with the C values of 7 to 10. So these are the species, plants with a very narrow range of habitat preference. So this was one metric that was statistically significant between the medians for attainment class A to B. So when we looked at the average buffer width by attainment class, and here on this graph, all the sites are visualized here. Well, the attainment class A had the highest minimum average buffer width, which was the minimum buffer width was 40 meters. There were numerous other wetlands with buffers that were greater than 40 meters. So the comparison of buffer width to an attainment class, this kind of gives you an idea of why it wasn't statistically significant. There just seemed to be a lot of overlap. So we reviewed the results for the water analyses. These are whisker and box plots for chloride and specific conductance. Once again, the wetlands with attainment class A stand out, while the others have values that overlap in range. So chloride and specific conductance were statistically significant between the attainment classes of A and C, because we did paired comparisons. So when we reviewed one of the other statistically significant parameters that surfaced was alkalinity. So they were significant between the predicted attainment classes of A and B. This was unfamiliar to me, but apparently researchers have attributed increases in alkalinity to acid rain, weathering of carbonates in bedrock, soils, and cement, such as bridges. So there is a human disturbance component that may be part of increases in alkalinity. We then use Spearman rank correlations to test the association between many of our variables. This graph depicts positive correlations as blue and tilted to the right, and negative correlations as red and tilted to the left. The darker the color and narrower the ellipse, the stronger the correlation. I have highlighted several correlations here between chloride and other parameters or metrics, and have provided the correlation values, the correlation coefficients. The strongest correlations are those that approach.
We're still seeing your alkalinity slide, so you can keep describing it. Unfortunately, we just aren't seeing the same thing, just so you know.
Okay, I was going to ask, but all right. Well, the next one is very colorful. You still on alkalinity?
Yeah. Oh, now we see your Spearman ranks.
Oh, good. Okay. All right. I'll start again. Okay. So we then use the Spearman rank correlations to test the association between many of our variables. This graph depicts positive correlations as blue and tilted to the right, and negative correlations as red and tilted to the left. The darker color and narrower the ellipse, the stronger the correlation. I've highlighted several correlations here between chloride and other parameters or metrics and provided the correlation coefficients. The strongest correlations are those that approach 1 or -1. The weakest correlations are those that are closer to 0. Among the stronger correlations observed were those between each rapid assessment and chloride. So you can see the ecological integrity assessment, there was a -0.83, and the wetland human disturbance was a 0.77. Correlations between 2 of the 3 GIS-based metrics from the EIA, the land use index and average buffer width, and chloride were also observed. So I thought that it was notable that just from a GIS basis, there was a pretty strong correlation between chloride and the land use index or average buffer width. While not a strong correlation coefficient, 2 fluorostic-based metrics are shown. The correlation between chloride and percent non-native species, which is 0.72, and chloride amine C, -0.52, were also noted. Some of the other coefficients in the table or ellipses are that are dark may represent items that may be autocorrelated. All right, so the next slide, and I'll wait till you all get there, is on macroinvertebrates and functional feeding group. Does the macroinvertebrate slide show up yet?
Yeah, we just got it, Sandy. Go ahead.
Okay. All right. So we looked at the functional feeding groups by predicted attainment class and observed that for the wetlands with attainment class A, the greatest difference among the attainment classes was the greater proportion of piercers. Among the piercers is the Corixidae, the water boatman. We had tried some ordination with the non-metric multi-dimensional scaling, and we weren't able to get as clear a picture of things as we had hoped. But we did determine that our macroinvertebrate samples were much closer correlated with Maine's samples, Maine's macroinvertebrates, when we did our second 2 years of sampling. So that was also an indication that maybe there was some issue with the first 2 years of our sampling. So the next slide shows some of the additional observations that we made. We documented, in our sampling, we documented plant species that were not yet documented in New Hampshire, such as Wolfia brasiliensis, which in the photo that you should see soon, if you don't see it now, when we observed it, it was like a layer of paint on the surface of the oxbow pond that we sampled. We documented new locations of rare species such as the pondweed Potamogeton zosteriformis and the Lemna trisulca, which is rare in New Hampshire. We also documented new locations of invasive species, including Potamogeton crispus, among others. There were some algal blooms. Some were green algae, such as Spirogyra. These were just collected, and the information was not part of any protocol, but we, when we saw something, we collected it and checked with some of the Watershed Management Bureau colleagues. We also, there was other cyanobacteria in another location, and we also found the nostoc colonies that look like peas. That was quite interesting. That is not quite associated with nutrients from what I understand. Found other wildlife. Picture here is the bryozoan colony, Pectinatella, that was found at one of our sites. Has everybody seen this slide?
Yes, Sandy, it came up.
Okay. All right. Getting to the end here. All right. So one of the challenges I think that we experience is I think sharing protocols is challenging. I think it's difficult to translate years of experience, like Maine's experience, sampling. They've been sampling since like 1998. I think they've done started with the pilot program. So to include these things in detailed protocols, I think is can be challenging. I think we found that we were sampling in very organic substrates, which was kind of a challenging setting to sample in. And sometimes we wanted guidance, but Maine was off doing their work. So it's difficult to get guidance sometimes when one needs it. And I think sometimes if you start working on a project and it goes different ways and you try different things, that kind of evolutionary process that leads to the protocols and model development carries with it a lot of information that may not be as clearly translated when someone else picks up protocols and goes forward with them. So considerations for next steps would be for rapid assessment of floristic quality assessment is continue the application of the ecological integrity assessment and floristic quality assessment, and grow the data set of vegetation data for FQA thresholds, including collecting additional data in non-exemplary wetlands. Our Natural Heritage Bureau tends to focus on the undisturbed exemplary natural communities, and so to get a better idea of what the range of human disturbance is, this is one of the major parts that this project, I think, contributed in terms of data. In terms of water quality, I think gathering additional data on chloride or specific conductance and evaluating that against buffer width in open water or other wetlands would be great to see whether some of the trends that we saw, observations carry through, or we could document them in a stronger way. We all know that buffers are important, but it would be great to have the data that really shows that that's local information. In terms of macroinvertebrates, perhaps targeting wetland type better. I know people typically will, in a study, will select a group or type of wetlands because then it's easier to analyze and make comparisons, but I think in terms of timing, we didn't have the universe available to really pick and choose when we were selecting our wetlands to sample. It would be great to examine any temporal effects of macroinvertebrate sampling. If you sample the end of June versus 3 weeks later in July, would that potentially change the attainment class that comes out? Analyzing macroinvertebrates by the specific location where sampled, while the guidance was to pick locations that were a comparable type. When you're in a wetland, some areas that look like they're comparable just are not. And it'd be great to see whether the specific samples taken from those individual locations provided information about that habitat. So in summary, we're not there yet in terms of identifying biocriteria. We have information about New Hampshire's wetlands that didn't exist previously for wetlands that represent a range of human disturbance. And then we've documented additional locations of rare and invasive species. We have built on our prior work, which brings New Hampshire closer to being able to select a method and establish biocriteria and thresholds to characterize wetland conditions. And I don't know what slide everybody's seeing right now.
Yeah, Sandy, we made it to your summary slide. So I think we ended with you.
Okay, well, I think I have 2 more slides, but they're just, one is with appreciation to the sampling team members and to the main DEP biomonitoring staff, which I have 2 slides, and then an acknowledgement slide to folks and to EPA for support for the wetland, through the wetland program development grants. And then there's a quote from Luna Leopold: "The health of our waters is the principal measure of how we live on the land." I think that's what we're trying to quantify. So I'll be glad to take any questions once you see all the slides.
Yeah, thank you, Sandy. They are all trickling through to us. There we go. We see Leopold's quote. So thank you so much for that presentation. We will move into a Q&A now. So there's 2 different ways that attendees can ask questions. One is you can send me, type in a question through the chat and questions feature of GoToWebinar, and just be sure to send that to staff so that I get that. You can also click on the hand icon to raise your hand and let me know that you want to ask your own question, or just unmute yourself and be sure to introduce yourself and ask your question. So those are your options to get me questions, and hopefully you have lots of them. Sandy, I'm going to let you stay on as presenter in case you want to try to go back to any slides if people have questions about something specific. It may take us 20 minutes to get there, so that may not work out. All right, Susan Jackson, do you want to go ahead and ask your question?
Yes, thank you. And thank you for the presentation. It's a lot of work. You seem to be able to distinguish between Class A and Class BC, a lump of BC wetlands. And I'm just wondering your thoughts right now. I know you've outlined a lot of work ahead, but do you think this is a matter of perhaps finding some different metrics that can distinguish between B and C versus A? Or do you think that's more perhaps the classification? And then I was curious, sorry, I'm packing in with questions. Has Maine had the same struggle in defining different or discriminating between different classes as you have with the B and the C?
Yeah, we have not, we didn't really dive into the model in detail to analyze the differences between them. Some of it, I think, is also because there may have been some sampling issues with the first 2 years of the data. So the A group actually has the fewest number of sites in the attainment class. So there were and the others have larger groups. We haven't dug into the macroinvertebrate data with that detail to figure out any of the differences. I had heard, and I don't know whether they still were finding it, that type of trend may be something that Maine has observed, but I don't want to speak for them, and that may have been something that several years ago was the case, but may not be the case now.
Thank you.
You're welcome. Thank you.
Thanks, and thanks everybody for sending in your questions. I will get to them next. I'm going to call on Charlie, and he's going to ask you another question. Hopefully this will work. Charlie, I'm going to try to unmute you. Oh.
Hello?
Yep,
Hi.
Go ahead, Charlie.
Awesome. Hi, this is Charlie from Vermont. Quick question, and I apologize if I missed this because I had to step out for a second, and it's kind of specific, but I was really curious. Here, too, we have some kind of preliminary data that seems to show a correlation between the chloride or the road salt type indicators and worse wetland condition. But what I've been trying to untangle is whether or not it's the road salt possibly affecting the wetland, or whether it's just that those correlate with wetlands that have roads going through them and the road itself is affecting the wetland. So I was just curious if you had noticed that or had any thoughts on that.
Yeah, that's an interesting question. Our buffer distance, the buffer distance in the EIA, in some places it could be road, in other places it may not be road. You know, it would, I don't know, a lot of these, I end up looking at it and saying, okay, where's the disturbance, at which end of the wetland, you know, which way you're thinking. You know, there's so much information there to try and dissect. It's a good question, but I don't know specifically. Haven't looked at the road versus disturbance or what have you.
Cool. Well, we'll have to keep looking at it too. Thanks. Very
Yeah,
neat.
sure.
All right, so I'm going to hit some of these questions in the order that they've come in. A question, is your report available yet that they could cite with these results?
The report will be finished soon, very soon, this month. So it will be available. Actually, there is a report for the first 2 years under the previous grant. There is a report available online, and I can, Audra, I could provide you with a link. Actually, if you Google NHDES wetland biomonitoring, I think that you'll probably come up in the first top ones too, because that's the way it's organized on our website right now. To the report from the first 2 years of work, which was 24 wetlands, and at that time we had, there were 10 that were considered indeterminate, and we actually, over the whole course of the thing, sampled 44 wetlands, made 44 trips, but the second time around we went back and sampled 2 of the wetlands again to get attainment class. But there is the earlier report on the website, and this report will be available soon.
Yeah, thanks, Sandy. I will send out that link to your website when I send out the post materials. But
Okay.
Thanks for directing. Size range of the wetlands in the study, and did you look at within wetland variants in macroinvertebrate samples, or were all 3 of the samples pulled together? For instance, the large wetlands might water quality vary slightly. Hopefully you understand that question.
Okay. Yeah, the samples were pooled together. And it was from that the generic diversity of 15 and total abundance of 50 numbers needed to be met for the model. The wetland size, I do have that information. Actually, in the first 2 years, the wetland size ranged from, we sampled a very small wetland that was like 3/10 of an acre to something that was really large, like 300 acres. The second 2 years, our wetlands were kind of in between those. Let me see, I'm trying to, okay. Yeah, so the wetlands over the 4 years range from almost a quarter acre to 330 acres. And we didn't field survey the entire wetland. We really focused on the aquatic part of it and the shoreline, and the GIS analysis included the full wetland system. And that's the acreage is the full wetland system.
Okay, thank you, Sandy, for that. Is New Hampshire using state or the regional COCs?
Good question. We actually have been using the state COCs. We started with the state COCs with the first group, with the first report. The new COCs hadn't been out. And then there were some questions about some of the invasives weren't, there were some questions about some of the values, not questioning the assignment of the specific value, but more that I think some of the invasives or non-natives were not given a value. I was trying to take this off the shelf and use it, use universal FQA to develop the values, and I sort of realized kind of late that I should have created my own database. And it can be loaded into FQA and you can use it yourself through that. And I sort of realized it too late. But I've used the state values, and the state values actually is what the Natural Heritage Bureau is continuing to use as well.
Okay, and then a question, if you could describe the methods that you used for the macroinvertebrate surveying and the taxonomic levels that you were using.
Okay, the taxonomic levels are generally to a genus. That's what the main model depends on. And the sampling, it's called a 1 meter measured sweep. And basically you would take the dip net and within a 1 meter length, kind of alongside the canoe was typically what we did. You would kind of bounce the dip net on the benthic surface in like 3 places, not totally picking it up in between, but picking it up a little bit. One of the problems was if you're in an area that has a really soft substrate, you end up with a ton of organic matter. So Maine has kind of a modified protocol for what to do in situations like that, and also it takes additional effort sieving out the organic material. But I think Maine's protocol is on their website as well, so someone could check there for to see their SOP.
Yeah, thanks. I will try to pull that up, and if I can find it, I will send that out to everybody. And a related question, if you think the staff who have an expertise in stream macroinvertebrates would be able to do the wetlands as well, if you feel like that's transferable enough.
I would think so. I'm actually, I have to admit, I'm actually a plant person. And although I had sampled macroinvertebrates in the past, I had really not done it in recent years. So it was kind of a newer thing for me. But we had training with Maine on a initially, and then after we had the problem with the organic material, we did another training. We took them to the same spot where we had the huge amounts of organic material, and that was helpful. So I think with adequate training, that someone who sampled in streams could do the sampling. We generally sampled from a canoe, but there were some sites that it would be easier. Either we couldn't get a canoe in there, or it would be easier to just sample in waders. So we, some of the sites we did that way.
A question, if you have a QAP or SOP available with the FQA methods, if that's online that people could find?
Yeah, I'll have to, let me check on the, there was a QAP, but I'll give you information.
Okay, and that was Carol Murphy in Rhode Island, so if you want to just connect directly with her, that might be easier too.
Okay.
You may not want to send that out to everybody, I understand. I'm just curious, going back to the chloride work that you're doing, or not that that's the emphasis, but the chloride aspect of your work rather, if you're trying to integrate that at all with, like, I believe New Hampshire has some TMDLs for chloride in the southern part of the state, if you're integrating with that work, or maybe talk about any other integration outside of what you're doing with the Heritage Program.
Well, one thing that I actually came across recently, but hadn't had the time to focus on, is there's been some work done by the UNH Stormwater Center on buffers and I think related to pollution loading and such. And I needed to take a look at more of the, I think they did a lot of literature reviews, but the information that I have here hasn't been integrated in anything else yet. I'll just leave it at that right now, but I think there is some good information that needs to be followed up on that could possibly be used in the TMDL or other areas.
Okay, great. And I just want to mention Beth Connors from Maine just sent a link to their methods. So some of you may be able to see that through the chat. Just wanted to point that out. And I'm going to just read her comment here. In Maine, we do both stream and wetland macroinvertebrate sampling. So yes, someone with stream experience could absolutely do the DipNet sweep. So thank you, Beth, for that input.
Great.
And I'm not seeing any other questions right now, but I'll just give it one more minute if people have anything else that they want to type in or if they want to ask a question. Thanks again to Sandy Crystall of New Hampshire DES Wetlands Bureau for presenting today, and thanks to you all for joining and participating.